Hello everyone. Today we'll be solving Cambridge International AS and A level Biology Paper 2 Structured Question, October-November 2021, Paper 2-2. There are two ways of cell prokaryote. There are two types of cell, prokaryote and eukaryote. Question number 1. There are two types of cell, prokaryote and eukaryote. Bacterial cells are prokaryote and plant cells are eukaryote. There are differences in the structural features that are common to bacterial cells and plant cells. For example, the cell surface membrane is a plant cell, contains cholesterol, but in a bacterial cell, the membrane contains molecules known as hapanoids. Cholesterol and hapanoids have the same function. So the function of cholesterol is to maintain membrane fluidity. So we can also say that the hapanoids also maintains membrane fluidity. Some of the main structural features common to both types of cell are shown in Table 1.1. Complete Table 1.1 by giving one difference between a bacterial cell and a plant cell for each structural feature that are listed. The difference between the cell surface membrane of the two types of cell has been completed for you. So cell surface membrane, the bacterial cell contains hapanoids and plant cell contains cholesterol. In terms of ribosome, the bacterial cell will contain 70S ribosome and the plant cell will contain 80S ribosome. DNA. In terms of DNA, the bacteria is going to have a circular DNA and plant cell is going to have a linear DNA. Eukaryotes have a linear DNA. Okay, cell wall. Bacterial cell wall is going to be made from peptidoglycan, whereas the plant cell wall is going to be made from cellulose. Part B. One role of the cell surface membrane of bacterial cell and plant cell is that transport of substances into and out of cells. Explain how membrane carrier proteins and membrane channel proteins are involved in the transport of substances into and out of cells. So basically, carrier proteins are needed for active transport and facilitated diffusion. Channel protein can provide facilitated diffusion as well. Carrier and channel protein are needed for the transport of ions and hydrophilic molecules. Carrier proteins are specific to the molecules that they transport, and they use ATP for active transport. Basically, we can say hydrophilic molecules, they are unable to go through the phospholipid bilayer, which is hydrophobic. And as a result, they use carrier protein and channel protein, which are a hydrophilic route through that partially permeable membrane. So most channel protein are not selective. However, aquaporin allows for passage of large quantity of water. Channel proteins are usually not selective. However, they will allow, for example, aquaporins will allow a lot of water to enter the cell. Guys, in my question, you can see I have used different colors. The colors indicate different marking point. And I have tried to make the answers as relatable as possible, according to what we have learned from the textbook and what our syllabus has taught us. All right. The way we should put those particular points in the marking scheme, I have tried to arrange them in that similar manner. So this red, the entirety of the red indicates 1 mark, the blue indicates second, then the green indicates 3 mark, and then again the process repeats with red, blue, and then green. So a total of 6 marking points are written over here. Writing any of the 3 points from here will get us the 3 marks. So it's a important thing that we write and memorize larger and bigger answers, so that in the end we can basically, even if we forget 1 or 2 points over here and over there, we can still write a pretty convincing answer so that we get the full marks every time. There are no negative markings, so it's a good idea to write to the point answer. However, write more points than necessary so that full mark is assured. Part C. Figure 1.1 is a photomicrograph showing the chloroplast in a plant leaf cells. We can see the chloroplast. Chloroplasts are found in the edges of the plant cell wall, as we can see. They are actually pushed by the central vacuole in this way, so that they are very close to the source of their carbon dioxide, and they can absorb carbon dioxide as fast as possible. Explain why the chloroplasts are seen only around the periphery edge of the plant cell. Chloroplasts are at the periphery because they are pushed by the vacuole in that particular position, should be a very good answer. Part D. Figure 1.2 shows plant cells in a root tip where cell division by mitosis is taking place. So we can see the prophase at this particular position. We can see metaphase. And these cells that we see over here, they kind of look like that they are in interphase. All right. And, in the interphase, they are probably after, they are either in the S phase or in the G2 phase because their nucleus size is pretty big. Okay. So this is prophase. And this is metaphase. The question says, identify two cells in figure 1.2 that are in different stages of mitosis. Draw a label line to each cell and add the name of the stages of mitosis that is shown by the cell. Okay, so we have done it. So let's move on to the next page. Question number 2. The high blood pressure at the arterial end of the capillary results in some components of the blood leaving the capillaries and forming tissue fluid. At the venous end, the presence of plasma proteins allow movement of water by osmosis back into the capillaries. Figure 2.1 is a diagram showing a capillary network. The lymph vessels and the blood vessels at the arterial and venous end of the network are shown. Okay, so basically when the blood is coming through the arteriole and then it is getting pushed into these capillaries, there is a higher pressure in these capillaries. There is a higher pressure. So what that pressure does is that it causes some of the plasma to leak out. It causes some of the plasma to leak out. Once it leaks out in between these particular body cells, we are going to call it tissue fluid. So in the blood, we're going to call it plasma. In the blood, we're going to call it plasma. However, once it leaks out, we're going to call it tissue fluid. And then once that tissue fluid makes its way into the lymphatic system, then we are going to call that particular fluid lymph. Okay. Some tissue fluid, they can make their way back into the venous end of the capillary, into the venous end of the capillary. They can go into that particular position. So part A, red blood cells and plasma proteins such as albumin remain in the capillaries and are not found in tissue fluid. Explain why red blood cells and albumin do not leave the capillary. So basically red blood cells and albumin. Albumin is a protein and red blood cells are cells. They are too large, so they cannot pass through the endothelial layer. Albumin cannot cross the endothelial cell membranes. They cannot pass through that endothelial cell membranes. They're just so big that they won't be able to pass through. Question part B. Name the chemical reagent or reagents used to test for protein in a sample of blood plasma and state the color change that would be seen if protein is present. Reagent or reagents. So basically we're going to need a biuret reagent. Biuret reagent tests for proteins, and if protein is present, then it will change its color from its initial color of blue to purple. Part C. When a person moves from sea level and stays at high altitude for a few months, there is an increase in the number of red blood cells per millimeter cube of blood. Explain why this increase occurs. Okay. So basically, when a particular person moves from sea level to a high altitude, there is a shortage of oxygen because percentage saturation of hemoglobin at sea level is almost 98%. However, when the particular person goes into a high altitude, the percentage saturation of the particular person's hemoglobin can be decreased to around 85%, depending to what altitude the particular person has gone to. So because of that, the body will try to adjust to it. So what the body is going to do is, in the beginning, the body is going to feel lethargic and like that, and then what's going to happen is that the low amount of oxygen in the blood is going to trigger a hormone, right, released from the kidney, known as erythropoietin. The erythropoietin hormone will then cause a higher amount of red blood cells to be produced within the body, and higher amount of red blood cells means that there will be more hemoglobin. And often people are prescribed actually high protein diet, and then they are also prescribed with more irons in their diet to cope up with it. The question says, explain why this increase occur. So basically the answer will be, at high altitude, partial pressure of oxygen is lower. So percentage saturation of hemoglobin with oxygen decreases. At high altitude, hemoglobin has a lower affinity for oxygen. More hemoglobin synthesized to compensate and help to transport the same quantity of oxygen as at sea level. And the EPO, which is like erythropoietin hormone, is secreted to increase the red blood cell production. Part D. A low blood albumin concentration can lead to a condition known as edema. Edema is a swelling of tissues caused by the accumulation of tissue fluid surrounding the body cells in the capillary network. Suggest and explain how a low blood albumin concentration can lead to edema. So a low blood albumin means that, it's like there is less solute present in the blood. So if there is less solute present in the blood, meaning the blood water potential is really, really high. So more water from the blood can enter the tissue fluid more than normal. So even though it enters the tissue fluid, it doesn't, from the arterial end, it enters the tissue fluid. However, in the venous end, the tissue fluid does not return back to the, back into the blood. And even it becomes difficult for the lymphatic system to take up that excess amount of liquid, which leads to edema. Part E. Albumin transports some cell signaling molecules from cells where they are synthesized to their target cells. The cell signaling molecules bind to specific cell surface membrane proteins on the target cell. Name the type of membrane protein to which cell signaling molecule binds. So it will be cell surface receptors. Cell surface receptors, they're going to readily accept cell surface receptors. They're going to readily accept the, whatever target, whatever signaling molecules, is targeted to that particular cell receptor. If the receptor matches, then it's going to bind with that molecule. Question number 3. Baculovirus is a virus that kills some of the insect pest of major crops. When the virus is released to the outside of the insect body, it is contained within stable protective structure known as polyhedrons. The main component of polyhedron is a protein molecule, polyhedrin. Polyhedrons can be sprayed onto plants as a bioinsecticide. They are ingested by feeding insect larvae, and once inside the insect gut, they break down to release the virus. Explain why the term infectious disease can be used to describe the effect of baculovirus on insect. So basically, baculovirus is a pathogen. We can see that it is transmitted from one host to another. And it is a disease because it is causing an ill effect on the insect. Part B, suggest and explain the conditions present in the insect gut that can cause the breakdown of polyhedrons. So basically, polyhedron is a protein. Polyhedron, we can see it's a protein molecule, all right, and it's made from polyhedrin. So polyhedrons is basically a protein. The insect gut can have highly acidic condition, which can disrupt hydrogen ionic bonds of the polyhedron proteins. Insect gut may have digestive enzymes, which can then break down the peptide bonds. The question says suggest, and then the question says explain. So we'll have to suggest whatever condition that is actually needed for digesting a particular protein, and write a solution to it, write an explanation to it. Part C. A polyhedrid molecule is composed of 3 identical polypeptides. Each polypeptide is 245 amino acid long. The first 10 amino acids of the polypeptide are shown in figure 3.1. We can see there is methionine, proline, aspartic acid, and so on. Figure 3.2a is a ribbon diagram of a single polypeptide. Figure 3.2b shows a polyhedrin molecule composed of 3 polypeptides. Okay, the two diagrams are not to the same scale. All right, we can see the both of the molecules. So we can see the complete molecule in part B, and we can see a single polypeptide of that complete molecule in part A. All right. Now the question says, with reference to figure 3.1 and 3.2, describe the structure of polyhedrin molecule. Okay, in this particular diagram, obviously we can see that the amino acid are joined one to with another, one with another. And so that is the primary structure. And then followed by that, we can see the alpha helix. So that is the secondary structure. We can see the beta pleated sheet. So that is the secondary structure of, it is formed by hydrogen bonds. And then since the molecule is folded in certain way, so there must be interaction between the R groups, which is basically the tertiary structure. So we can answer this question in this way. The primary structure consists of a sequence of amino acids that are joined by peptide bonds. The secondary structure consists of alpha helix and beta pleated sheet, which are basically formed by hydrogen bond between the CO and NH. The tertiary structure has interaction between side chains, example, hydrogen bonds, ionic bonds, disulfide bridges, and so on. The quaternary structure, example, the 3 identical polypeptides that are joined together over here, all right, they are interacting with each other. They are interacting with each other by means of hydrogen bond between the R groups, ionic bonds, disulfide bridges. So we can write those in the answer. Part D, the sequence of DNA nucleotides for the gene in baculovirus that codes for the polyhedrin polypeptide has been determined. Explain why the amino acid sequence polypeptide cannot be used to deduce the sequence of the nucleotides in the gene that codes for the polyhedrin molecule. Okay, you see, amino acid can be coded by more than one codon. There can be multiple codons that can actually code for a single amino acid. And since there are 64 codons only for 20 amino acids, the third base in the codon can be very different. Usually the first 2 bases matches, and then the third base is different. Stop codons do not specify any amino acid. So we want to know the stop codon. But there are more than one triplet base for stop codon as well. So that's why we're not going to be able to know it. Question number 4. Berlinka is a variety of common grapevine. Vitis vinifera. Berlinka grapes are used for making wine and sold as fruit. There are economic and ecological benefits from using less water to irrigate grapevine plants while producing high crop yield. The rate of flow of sap within xylem vessels from roots to leaves can be used as an estimate of the rate of transpiration. This also indicates water uptake. Part A. The hydrogen bonding of water molecule is important in the transport of sap within xylem vessels. State the terms used to describe water molecules sticking together within the xylem vessel. So water molecules sticking together within the xylem vessel will be cohesion. Water molecules sticking to the cellulose molecule in the lining of the xylem vessel. All right, so basically when water molecules stick with the cellulose molecule, we call it adhere. So it adheres to it. So it's going to be adhesion. Part B. An experiment was carried out to investigate the effect of leaf area on the rate of flow of the xylem sap during fruit development. The flow rate was measured over a 3-day period in 3 Berlinka grapevine plants with different total leaf area. The plants were growing in the same condition. The results are shown in figure 4.1. Leaf area for grapevine 1/13.7 meter square. Okay, so we can see this is the one. Okay. All right. And then similarly, leaf area for grapevine 2 with less and less. We can see with the decrease in surface area in terms of, we can see with the decrease in surface area, we can see also the flow of xylem sap also decreases significantly. Okay, let's carry on. Part 1. The general pattern of results in figure 4.1 is the same for the 3 Berlinka grapevines. Explain why general pattern of results is the same. So basically, during the day, the rate of transpiration is going to be higher. That's always the case. And at night, the rate of transpiration is going to be lower. Stomata are open more during the daylight to obtain carbon dioxide, which is needed for photosynthesis. And all 3 vines were kept in the same condition. So a change condition that affect transpiration of one also affect for all the other 3. Part 2. With reference to figure 4.1, explain the differences in the results between the 3 grapevines 1 to 3. So as we can see, the grapevines 1, 2, 3 has, grapevine 1 has 13.7 meters square of leaf area, and then 9.6 and 3.2. So with the decrease in total leaf surface area, all right, there is a decrease in transpiration. And the greater the total area of leaf, obviously there will be more number of stomata. Grapevine 1 has the highest transpiration rate and highest number of stomata, whereas the grapevine 3, we can see it has the lowest surface area and lowest number of stomata, and as a result, lowest transpiration rate. Part 3. Suggest an explanation for the decrease in flow rate of xylem sap shown at time P in figure 4.1. So an explanation for the decrease in flow rate of xylem sap. So we can see, if we look at the graph, at particular point P, something happened which caused the xylem to flow. Less amount of sap was flowing through the xylem at this particular point of time. It can be due to high humidity, all right? It can be due to rain. It can be due to cloud. So we can write down, maybe the situation was cloudy, maybe there was rain, or we can write there was high humidity. Since the question says suggest, so any of this answer will be accepted. Part 4, outline how you would determine the surface area of one side of a leaf. All right. So the leaf is placed on a square graph paper. And then, we can draw the leaf on that particular graph paper. And then the number of full squares, or the squares, those that are full more than 50%, can be counted. Squares, those that have less than 50%, does not need to be counted. And in that way, if we count the graph area, surface area, then we can get the surface area of the leaf as well, an estimate of the surface area of the leaf. Question number 5. The causative organism of measles is morbillivirus. Young children who have not been vaccinated for measles are at highest risk of becoming ill and of developing complication associated with the disease. Part A. The genetic material for morbillivirus is a strand of RNA. Statement A, B, C can relate to the structure of RNA. State the correct term or terms to match each statements A to C. The names of the 2 purines and 2 pyrimidines in RNA. All right, so the purines are going to be same as DNA, so they're going to be adenine and guanine. However, the pyrimidines will be uracil instead of thymine, and it's going to be cytosine. B, the type of covalent bond between RNA nucleotides. You see, RNA nucleotides have phosphodiester bond between them. Phosphodiester bond between them. Part C, the pentose sugar of the RNA nucleotide. The pentose sugar of the RNA nucleotide is a ribonucleic acid. So it's a ribonucleic acid, so ribose. Part B, describe how measles is transmitted. Measles is transmitted from an infected person by droplet infection. So when an infected person breathes out, coughs, or sneezes, when the airborne droplets is breathed in by uninfected person, measles can be spread from infected person in that way to an uninfected person. Measles can also be transmitted when uninfected person touches, like after sneezing, touches the hand, and then basically the hand touches a particular surface of a railing or anything. Then when the uninfected person touches that infected surface and puts his hand in mouth or nose or into the eyes, then the particular person can get infected. C, young children with measles may develop difficulties with breathing. This is made worse if the child is continually exposed to tobacco smoke. Tobacco smoke contains harmful compounds such as carbon monoxide. Inhaled tobacco smoke passes through the larynx, voice box, and other structures in the gas exchange system to reach the alveoli. Complete figure 5.1 to list the correct sequence, the main structure of gas exchange system through which the tobacco smoke passes to reach the alveoli. So of course, once the tobacco smoke, or generally when air is breathed in, it goes from larynx to trachea to bronchus to bronchiole. So the tobacco smoke will also go from larynx to trachea and then from bronchus to bronchiole. So first is going to be trachea, and then it's going to be bronchus, and then it's going to be bronchiole. Next question. The question says, state the short-term effects of carbon monoxide on the cardiovascular system. The effect of carbon monoxide. Carbon monoxide binds with hemoglobin to form carboxyhemoglobin. So as a result, less hemoglobin is available to bind to oxygen. Because once the hemoglobin forms carboxyhemoglobin, it can no longer bind to oxygen or carbon dioxide or anything. Carbon monoxide reduces the percentage saturation of hemoglobin with oxygen in this way, because the hemoglobins are busy carrying the carbon monoxide and they are irreversible. So as a result, less quantity of oxygen is transported to active cells. Part D. Smallpox is a disease that has been eradicated with the help of global vaccination program. Measles has not been eradicated even though there is a global vaccination program. Figure 5.2 is a graph showing the estimate number of deaths from measles globally between 2000 and 2017 for people who have not been vaccinated. Okay, so we can see from 2000 to 2017, there is a steady number of deaths from measles increasing. State the trend shown in figure 5.2 between 2000 and 2017. We can see that there is a steady increase in the number of estimated deaths from measles from 2000 to 2017. Part 2. Suggest reasons for the trend shown in figure 5.2. So by the way, the trend shown was that there was a steady increase in estimated number of deaths from measles from 2000 to 2017. Basically, it can be because as the global population has increased, the higher the number of unvaccinated people has increased as well. An increasing number of people with HIV, all right, and those people, those that got infected, since they're HIV positive, they got infected with measles, they were also spreading the measles more, and less herd immunity due to increase in number of people living with HIV that can, spread the measles. Risk of death for measles is higher in unvaccinated people, and there is also a difficulty in obtaining the vaccine in remote communities because the vaccine is very hard to carry. It has to be temperature controlled and maintained. And finally, only highly trained professional nurse are actually able to administer the vaccine. So it's a difficult thing to do. Part E. A child with leukemia is at high risk of developing measles even though the child has a high white blood cell count and has been vaccinated against the disease. Explain why this child is at high risk of developing measles. You see, even though a person, those who have leukemia, have a high count of white blood cells, the white blood cells is due to uncontrolled mitosis of white blood cells. So they're actually not functional, all right, to provide any immunity. And due to the weakened immune system, they are more likely to develop measles. Those, people, those who are suffering from leukemia, they will, get the measles, and vaccination is less effective on them because it gives a very weak immune response. Their immune system is already weak because of the leukemia. Part 6. Amylase is an enzyme that catalyzes the hydrolysis of starch into reducing sugar. A student carried out an experiment to investigate the hydrolysis of starch using immobilized amylase. Figure 6.1 is a diagram of the apparatus that was used in the investigation. We can see the starch solution present over here. When the tap is open, the starch solution will go through the immobilized amylase, through the alginate beads of the immobilized amylase, and then the product will be collected in this beaker. The alginate beads were all the same size. Both taps were opened to allow the starch solution to flow down the column and for the product to be collected. The product was tested for the presence of reducing sugar and starch. The results of the investigation showed that the product collected in the beaker contained reducing sugar and starch. With reference to figure 6.1, describe a method that would allow the student to use the immobilized amylase to collect a product that contains reducing sugar only. Okay, so if the student wanted to collect reducing sugar only, then the student should open this tab while keeping this particular tab closed and allow this particular get vessel fill with the starch, and the starch will then be broken down. All right, the starch will then be broken down into reducing sugar once enough amount of time has passed, which depending on what amount of enzyme has been added, let's say 5 minutes or 10 minutes or 15 minutes, then the student can allow the bottom tab to open and the liquid to come out. And as a result, this the liquid is going to contain only reducing sugar. So this is a plausible answer that we can write. The student keeps the top tab open and the bottom tab closed to allow the full hydrolysis. And then a product sample is tested for the presence of starch using iodine solution. The student can also use smaller beads to increase the rate of hydrolysis as well. One standardized variable in the investigation is the size of alginate beads. Suggest one reason why using a larger or smaller alginate beads in the column would affect the results. You see, if alginate beads, the size will affect the surface area to volume ratio. All right, if the size is increased, then the surface area to volume ratio will decrease. So larger beads will slow the rate of diffusion of the starch into the beads and thereby their breakdown as well. Okay, guys, that's all for today's class. We're done with this particular paper, Winter 21, paper 22. And, if you want us to see, if you want question papers like this to be solved, all right, and you want to get the answers like this, subscribe and stay with our channel. We are thinking of uploading one video every day, and we will try to cover up question papers, those that are related to chemistry and biology for any level of IGCSE, GCE, all right, O levels and A levels. Thank you. Bye bye. See you in the next class.